Hello, everyone. It is my great pleasure to introduce to you a distinguished scholar, a researcher, and of course, a friend, Dr. Dobromir Rahnov. Dr. Rahnov received his PhD in psychology from Columbia University and completed his postdoctoral fellowship at UCA Berkeley. He joined Georgia Tech University in 2015, where he's currently Blanchard Early Career Professor. He uses a variety of methods such as fMRI, TMS, psychophysics, and computational modeling to study perceptual decision making. His work appears in high-impact journals such as Behavior and Brain Sciences, TNAS, Nature Communication, Nature Human and Behavior, and many more. And he received multiple grants from NIH, NSF, and Office of Naval Research. Dr. Rahnov, thank you for being here. And without further ado, I will let you take it from here. Thank you.
Thank you, Armin. Thank you for this nice introduction, and thank you so much for inviting me.
Of course.
So today I want to tell you about some of our recent work. Pretty much all of it is either just freshly accepted or still in preparation. So I am really looking forward to questions and comments, and we're actively working on all of this. So it'd be great to see what everybody has to say. So just as a quick introduction, my lab overall works on perceptual decision making. And in perceptual decision making, the big question that we're trying to answer is, how do people make decisions based on ambiguous and noisy visual information? You can think of this in the real world. If you're driving, for example, in a fog condition, estimating the distance to the car in front of you is probably going to be a lot more difficult than if you're driving in perfectly clear conditions. In the lab, we tend to study these kind of questions using stimuli like the dot motion stimuli or some more complex designs. But we're trying to get at that same fundamental question. Now, in the lab, we take several different approaches. We use psychophysics, cognitive modeling, we study the neural bases, and we also have recently started to use artificial neural networks. Today, I'm mostly going to focus on the neural basis component, but also have some cognitive modeling and artificial neural networks sprinkled in there. The psychophysics, I don't think this is the right audience for it, so I've left it for another doc. So the question that I want to discuss today is relates to the idiosyncrasy of brain activations. So we can ask a general question, which is, how do you find the neural correlates of a behavioral outcome? So if you're studying, for example, reaction time or accuracy or confidence or anything else, how is it that you find the neural correlates of that? This seems like a deceptively easy question. We're doing that all the time. We've been doing that as a field for many decades. And the standard way of doing this, so if you permit me to just show you a screenshot of the SPM control panel. You start with pre-processing, then you have a first level analysis that is studying the individual. And then immediately afterwards, you have a second level analysis. This is the group. And for the most part, a lot of our research, by definition, just looks at the second level group analysis. And when we talk about the neural correlates of something, what we are talking about is this map that we're going to get at the second level. But what we do is usually we just ignore the variability in the maps here. So we assume that the variability is mostly noise or not very meaningful, or at least not very interesting. And we just jump to the second level, see what is consistent across subjects. This is a perfectly fine approach, but there's also the question, are we missing important things at the first level? So to answer that, we need to ask ourselves, how different are subjects from each other? So we know that people are not all the same. There's going to be some meaningful difference, but how different? Is it that they're mostly the same? Maybe some small differences? Are they mostly different? A little bit of is the same? Is it somewhere in between? We need to be able to quantify that in order to really understand what is the most useful way of studying brain activations. Should we be exclusively focusing on the second level, or should we be paying a lot more attention to the first level? So here's how we have approached this question. For this talk, I think all or almost all of the data will come from one specific experiment. It's a perceptual decision-making task. It starts with a fixation dot. Then people see this stimulus that consists of a cloud of blue and red dots. People need to make a decision. The decision is which is the dominant color. So are there more blue dots or more red dots? And then they need to give a confidence rating. It's a really simple task. The good thing about it is that these trials go really fast. So what we can do is we can collect a lot of trials per subject. So in this experiment, we have 50 subjects and 768 trials per subject. And I want to mention this is work of my, unfortunately, former postdoc. As of last Friday, he moved on to a new position with the Army, Johan Noguchi. So people complete this task, and then we're going to look at the brain maps for different people for different types of contrast. So I'm going to call these single subject activations. So let's start by just looking at 3 subjects, just literally the first 3 subjects that we collected. This is the task-based activation, meaning just task compared to rest. And this is what you get for subject 1, subject 2, and subject 3. And you can see that there's a lot of similarity. And there's this strong activation in the visual cortex, which makes sense because there's visual stimuli. There's some other similarities, but there's also some differences. We can look at the reaction time based activation. So this is just a contrast of fast minus slow responses. And again, there's some similarities, mostly visual cortex again, but some differences as well. And finally, we can also look at confidence, which is again a contrast of high confidence minus low confidence. And again, the same kind of thing appears. So there's some similarities and some differences. So we can highlight some areas, very strong consistency. For example, the visual cortex here. There's negative activation in the parietal cortex here, and there's positive activation in the prefrontal cortex here. But we can also highlight differences. So for example, if we look at this motor area, we see negative activations for subjects 1 and 3, but positive activation for subject 2. This slightly different motor area, same thing is happening for reaction time based activations. And for confidence, here we have subjects 1 and 2 showing negative activations in this prefrontal area, but subject 3 showing positive activation in that same prefrontal area. Now, obviously, we can just play this game. You give me any 3 activations, now find something that is similar and something that is different. The question is, how consistent is that? Is this just noise? Or is that something that is consistent, something that is really signal? So this subject is fundamentally different in their prefrontal activation compared to these 2 other subjects. So how do you do that? Well, one approach is you look at the odd and even trials separately to get a sense of the consistency of the activations for each subject. So if we now look at the odd trials, the map for the odd trials and the map for the even trials, we see that the maps are similar but not identical. Because of noise, they won't be identical. Same thing for subject 2, same thing for subject 3. We can plot these for the reaction time based activations and for the confidence based activations. And if I now highlight those same areas again, there seems to be a good amount of consistency for these positive activations, which is something that we may expect. But there also seems to be consistency in these differences. So subjects 1 and 2, subject 1 is showing negative activations for both odd and even, and so does subject 3, but subject 2 is consistently showing positive activations in this area. Same thing for this other motor area, and same thing for this area in the prefrontal cortex. But still, this is just very qualitative. We need to figure out how to make this more precise, more quantitative. So how do we do that? And just to summarize, we find some similarities between subjects, some differences between subjects, nothing particularly surprising at this point. The question is how to quantify what is common and what is different. So here's the approach that we took. We are going to start by computing what we call within subject reliability. So we're going to take subject 1, we're going to take the maps for the odd and the even trials, and then we're just going to correlate voxel by voxel the correlation of the activations for the odd and the even maps. I'm going to call this R1 for subject 1. Then we're going to do that for subject 2 and all the way to subject 50. Then we're going to take these 50 R values and we're going to average them. So by averaging them, we get the overall within subject reliability of the data. Okay, so now we have an estimate of the within subject reliability. Now we also need to compute what we call a subject to group similarity. How are you going to do that? We're going to take subject 1, the map for subject 1, subject 2, subject 3, subject 4, subject 5, all the way to subject 50. And then we are going to correlate subject 1 to subject 2, subject 1 to subject 3, subject 1 to subject 4, and so on and so forth. So this is all for subject 1, but then we're going to compute all of these correlations for each subject separately. So then subject 2 to subject 3, subject 2 to subject 4, and so on and so forth. You get all of these R values that are pairwise correlations of pairwise brains. And then you can again average all of these to obtain an overall subject to group similarity. Everybody with me so far? Okay, simple enough. So what do you get when you do that? Well, in our data set, we let's start by plotting the reliability. So these are the reliability values that we get. This is about R of 0.7 or 0.8. And we're going to just make things more robust, we're going to examine this for smoothing level of 5 millimeters, 10 millimeters, and 20 millimeters. For analysis where I'm not showing the smoothing level, we're always using 10 millimeters, but the results stay the same for all the smoothing levels. Okay, so this is pretty good reliability, not perfect, pretty much what you can expect. One of the things that we were worried about is that maybe there's a lot of noisy voxels that just don't really meaningfully contribute to the activations. So maybe we want to exclude those. So what we did was we additionally looked at tight and tighter selections of voxels. So this is 100% of voxels selected, meaning we're analyzing the whole brain. But we can also just look at the 75% most robustly activated voxels, either positively or negatively activated, and then just look at them and do all of the correlations based on them. Do that for the top 50, top 25, top 10%. And as you can see, the reliability slightly increases as you select the most reliable voxels. Okay, so this is the within subject reliability. What about the subject to group similarity? This is what that looks like. As you can see, the values are slightly lower, but they're still quite high. So people are consistent with each other, but they're not perfectly consistent with each other. Again, something that you may expect. The question is, how do you compare these values? So this is for task-based activations. The same thing happens for reaction time-based activation. Again, slightly lower reliability, but still pretty good, and slightly lower subject to group similarity, but still pretty good. And as you can see, higher smoothing results in slightly better values compared to lower smoothing. This is perhaps due to small level differences between the realignment, but we're not 100% sure about the interpretation here. And the same thing happens for confidence. You get slightly lower reliability values and also slightly lower similarity values. Okay, so far so good, but this doesn't help us too much. We can see decent reliability, decent similarity. That's not going to get us all that far. So the critical thing that we did was then we create a computational model. So the computational model is really the heart of this project. So let me try to explain how this works. What we want to do is we want to simulate what we call idealized beta values. So we're going to simulate each of the voxels in the brain. Let's say we've collected 70,000 voxels in the brain. We're going to simulate each of the 70,000 voxels in the brain. These are going to be new, random, independent 70,000 voxels. So we're not going to use any of the information that we have from real brains. We're just simulating 70,000 random voxels out there. The way we're going to simulate that is we're going to use two types of factors. One is group level factors. So this is the task, the RT, and the confidence. And these factors will be the same for each subject. And then we're going to have subject level factors. This again, the task, the RT, and the confidence, but for each subject separately. So now the value of this factor is going to differ from subject to subject. It's going to remain the same for all trials within a subject, but it will differ from subject to subject. And finally, we're going to have a noise factor. The noise factor will differ not only from subject to subject, but also from trial to trial. So this is going to just give random noise on each trial when we're simulating these idealized beta values. Okay, so the critical thing here is that each of these factors will have some weight. So we have 7 factors and 7 weights, but we can, without loss of generality, we can set one of the weights at 1. So we're going to set the noise weight to be 1. And then we just need to estimate the 6 remaining weights. The way we're going to do that is we will take the same within subject reliability values, the mean values across all of the subjects, and the mean subject to group similarity values, again, across all subjects. And we're going to adjust these 6 weights. We're going to do model fitting on these 6 weights until we can get fits that are as good as possible. Okay, so can we do that? Using this kind of simple model with only 6 free parameters, can we fit the within subject reliability and the subject to group similarity values? Overall, we can do that pretty well. So here in red is the data that I had shown you previously, and in blue is the model. This is just for the 10 millimeter smoothing, but we did it separately for her 5 and for 20, and the results are very similar. So this is not a perfect fit, but it's a pretty decent fit. And I just want to note that we are only fitting to this value, to the 100%. So everything else is sort of coming out naturally from the model. This is what we get for RT-based activation, and this is for the confidence-based activation. This is the only place where there's a slightly larger deviation between the model and the data. But in any case, we only have 6 free parameters, so overall this is a pretty decent fit. Okay, so we can fit, and we have a simple model, so that is great, but that by itself is not super interesting. Okay, you have a model, so what? What is the most interesting is then to go back and to look at the weights. So how large are the weights for the group level factors versus the subject level factors? Again, we're starting with the noise level factor having a weight of 1, and the width here of the lines will be proportional to the weights. So what do we get for the task factors? They're way smaller than the noise, first of all. The noise is just much stronger than the task factors. But importantly, they're about equal. So the group level factor and the subject level factors, they're about equal. For RT, we get even lower values, but still they're about equal. The subject level factor is slightly larger. And for confidence, the same thing, still lower. And again, the subject level factor is slightly larger than the confidence, the group level factor. Okay, so these values suggest that to fit the data, to reproduce this pattern of results we're observing, we get stronger subject level factors compared to group level factors. We can make this more quantitative by looking at the 5 millimeter, 10 millimeter, and 20 millimeter separately. And we're plotting it here. These are the raw weights. But the most interesting thing is to look at the ratios. So when you look at the ratios, these are the ratios that you get for the task. So they're sort of spread out right around 1 for a task. And they're close to 1, but maybe slightly larger on average for RT and confidence. And you can also look at the total percent of weight. So noise dominates everything. So at least in the experiment that we collected, noise is the primary thing that is driving the activations, unfortunately. But beyond that, there's the group level factors and the subject level factors. And the subject level factors are slightly larger than the group level factors. Okay, to summarize, so far, we have now built a simple computational model that allows us to estimate the contribution of group versus subject level factors in brain activations. And we applied it to this very specific data set, but the model is completely general. You can take anything that you collect, any kind of data set, and if anything, if you don't have confidence in reaction time, you'd only have the task activation, so you can just have 2 free parameters to fit, and it will be a very simple model, very easy to fit. So what I showed you is for the standard perceptual analysis, just test, task, or RT or confidence. The subject level factors have similar weight, maybe slightly larger than the group level factors. Okay, so far so good. But you may ask, are there some other analyses that change this ratio? So is there some other analysis that we can run that will give us different results, different subject to group factor ratios? So another thing that we did was to run the same analysis, but at the block level. Now, what does that mean? Our experiment was designed to be, so it was structured in blocks of 8 trials. So this is a single trial, each block has 8 of these. So then what we did was we computed the average confidence and the average RT for each block. And then we ran the same analysis, but comparing blocks with high RT versus blocks with low RT, and blocks with high confidence versus blocks with low confidence. It is not a standard type way of looking at data. We don't usually look at fast blocks versus slow blocks, and blocks with high confidence versus blocks with low confidence. But it's nothing fundamentally that fundamentally prevents us from doing that. So then what do we find for task-based activations? This is subject 1, subject 2, and subject 3. And this is the odd and even activations underneath. And you can see that for the task, it is very similar to what we saw before. There's a lot of similarities, some differences, looks quite similar. But for RT, we're actually getting something very different. So this is the activation for subject 1. This was the activation for subject 2. This is the activation for subject 3. And you can see that these are pretty consistent within a subject, but really different across subjects. I mean, there's this really strong activation in the visual cortex here and a very strong negative activation in the visual cortex. And you can see all kinds of differences across subjects. Same thing for confidence-based activation. Again, things are quite consistent within a subject. So these look quite similar, but they're really different across subjects. So this already suggests that in this analysis, we must be getting a lot stronger subject level factors compared to group level factors. So we can again plot the reliability, the within subject reliability, and what we see is that we have really strong reliability for the task activation when we compute it based on all of the data, and lower ones for reaction time and confidence. This is why we also look at the task-based activation for when we only look at 2 blocks to sort of match the reliability between the task and the reaction time and confidence. So then you still see some level of reliability, but now much lower because we have thrown away most of our data. We have only kept 2 blocks. Then when you look at the subject to group similarity, now you see this pattern where subjects are really different when it comes to reaction time and confidence. And they're somewhat different, but a lot more similar when it comes to the task, regardless of whether we're looking at the 96 or the 2 blocks. So we can then apply the same model to these data. So we're going to fit it in exactly the same way with exactly the same factors. And what are we going to get? So this is the noise level factor of 1. Now the task actually has a lot stronger weight. So this is why block level analysis are really quite robust, because these are a lot stronger ways than what we were getting before on the trial by trial data. But when we look at the reaction time and confidence, we see really low values here and actually decently high values here. So the subject level factors are a lot higher compared to the group level factors. So again, these are the raw ways that we got, and these are the 3 levels of smoothing. And when we look at the ratios are slightly larger for the task, but they're really large for the reaction time and confidence. So all of these values are above 6 and with a mean of about 8. So the subject level factors are 8 times stronger for this type of analysis compared to the group level factor. And is the total percent of total weight explained by each of these factors. So I showed you activations. We can do the same because we're working on the block level. We can look at functional connectivity instead. So we can look within a block and compute, rather than just activation, we can compute functional connectivity. And we use the Schaefer atlas of 200 by 200 regions. So this is the correlation matrix that you get. The Schaefer atlas is divided in 7 communities: the frontoparietal network, the default mode network, dorsal attention, limbic, ventral attention, somatomotor, and visual. And you see one pattern of activation. You see so mostly similar pattern of activation for subject 2 and mostly similar pattern of activation for subject 3. So this is for the task, but for reaction time, you're going to see something very different. Now, subjects look quite different from each other. For example, you can see this mostly red area here, and that becomes mostly blue for this subject. And same thing for confidence, a lot of differences between the subjects. These do not look all that similar to each other. So when we look at the same kind of results within subject reliability versus subject to group similarity, we see a similar kind of thing where the within subject reliability is high for the task when we look at all 96 blocks, and gets lower for this RT and this confidence. I'm sorry, the label is not there. But things change when we look at subject to group similarity. So the task this is from the 96 blocks, this is just from the 2 blocks, has much higher subject to group similarity compared to the RT and the confidence. So this just confirms what we saw from this for this same analysis when we look at just the activations. So to summarize here, we already talked how we have a simple computational model, and then for standard perceptual analysis, the subject level factors have similar weight. Now, for this non-standard block-based analysis, we show a very different. We see a different result where for the task is still similar than before, but for reaction time and confidence, we see really strong idiosyncrasy. And this happens both for the activations and for the functional connectivity data. All right. So either way, regardless of whether you're looking at this more standard analysis or this more different analysis, the subject level factors seem to matter a lot. And we're gonna now go back to just looking at the trial level analysis, but we're gonna start looking at decoding for individual subjects. So we want to look at, given that there's a lot of differences between subjects, given that there's a lot of idiosyncrasy, can we predict reaction time and confidence, but not across all subjects, but look at individual subjects separately? So what we're going to do is we're going to divide the brain into 200 regions using the Schaefer Atlas, like before. So this is what the Schaefer Atlas looks like. Again, it has 7 communities. And then for each ROI, so for each of the 200 ROIs that we get from the Schaefer Atlas, we're going to test if we can predict reaction time and confidence, and we're going to do that for each subject separately. Okay, and then we're going to look at significance within each subject individually. So here's how we're going to do that. So we take a subject, we take all of the trials, and we're going to randomly select half of the trials that are colored in red and half the trials that are colored in green. So we're going to do a random split into 2. Then we're going to take the first half here, and we're going to train a classifier, just a linear regression, to predict the behavior, so whether it is reaction time or confidence, based on the activation. And we get some sort of a relationship. In this case, let's say it's a negative relationship. Then we're going to apply this to the activations that we get in the remaining trials. So based on these activations, we're going to predict behavior. So this is predicted behavior here. And finally, we're going to correlate the predicted behavior with the actual empirical behavior that we observe. And we're going to get a correlation coefficient, let's say R1. We're going to repeat this 25 times to make this as robust as possible. So 25 different splits. And we're going to get 25 empirical R values. And to be able to make a comparison, we're going to do a random shuffle and redo this whole process 1,000 times. So this is to create a now distribution that would allow us to then compute a p-value by comparing the empirical 25 values to this random distribution of the now distribution obtained by the shuffling. Okay, I don't see faces, but hopefully this makes sense. This is a very standard way of doing things. So how well can we predict response time? So this may not surprise you, but we find significant p less than 0.05 uncorrected decoding for at least one subject for all 200 ROIs. So for any of the 200 ROIs, when we look at decoding, we can find at least one subject where we have predicted reaction time significantly. Now we have 50 subjects, and by chance you'd see that in one of 20 subjects. So this is not particularly surprising. But what we can then do is we can actually correct. So we're going to do bonfire correction because we're doing 50 comparisons. So for a given ROI, we have 50 subjects. So we have 50 options to get a significant result. So we're going to do bonfire correction for 50 comparisons and then see for how many ROIs, how many of the 200 ROIs we can still predict significantly for at least one subject. So I should have included a poll maybe here to get a sense of people's intuition, but here's what we find. We can predict at least one subject significantly after bonfire correction for 186 out of 200 ROIs, or 93%. So for the great majority of the brain, we can significantly predict reaction time for at least one subject after doing a very conservative correction for multiple comparison. This is what the predictions actually look like. This is without correction, and this is with correction. So you can see that for many ROIs, like for example, in the DA, in the dorsal attention network, the prediction is mostly in one direction. So this is mostly positive relationship between activation and RT. But in some cases, like the visual cortex, for example, you see a lot of idiosyncrasy. Some people have positive, some people have negative activations or negative relationships. And remember, this is bonfire corrected. So this is really consistent results that just naturally just go in the opposite direction for different people. We can do the same thing for confidence. And again, we find that all 200 ROIs can be predicted without any correction. Again, this is not super surprising. When you do correction, it's a little bit less, but it's still a lot of the brain. 157 out of 200 ROIs, or 78.5% of subjects show, of ROIs, for that percentage, for 78.5% of the ROIs, we can predict the confidence for at least one subject after bonfire correction. And this is the pattern of decoding without correction and with bonfire correction. And here you see a slightly different pattern, or quite different. So visual cortex is mostly negative now. But there's other networks, like for example, the DAN, the dorsal attention network, is now quite mixed. So some subjects negative, some subjects positive. We can, just for comparison purposes, we can do the standard second level decoding. So do the same prediction that we had already done for each subject separately, but then do the analysis not on each individual, but just look at the consistency across all 50 subjects, which is the way usually we do decoding analysis. And if you do that, you're going to see that uncorrected, you can only predict a little more than 50%, and after bonfire correction, you only predict RT from 12% of the brain. And this is what this looks like. These are the regions that survive bonfire correction. And for confidence, you only predict for 3.5% of the brain, and this is what this looks like. So these are the kind of analysis we're used to. So we're used to seeing 3.5 for up to 12% of the brain activated, and we make a big deal out of those regions as being fundamental about the behavioral construct. So we think that those 12% of regions are fundamentally important to RT, and these 3.5% of regions are fundamentally important to confidence. But we actually ignore the fact that most of the brain is important, or at least is associated strongly, very strongly, with RT for at least one subject, and for confidence for at least one subject. Now, in this experiment, we have a lot of data. So what we could do is we could also look at how many trials do you need for predicting behavior. So we only use 700 of our trials because some subjects didn't have the full 768. And we tested a different number of trials, which is on the the first row here with percent of trials. And so we would predict from 35 trials and then we'll build the model based on 35 trials and then predict the other remaining 95% of trials. So you can see that this is the prediction value that we can get. It shoots up at around 350 trials in our data, and this is the red is the standard deviation. So you want relatively low variance. So this is what we were using 350 trials in our data, and you can see that this works quite well. But if you have a lot less trials, this may not work at all. And same thing for confidence. You bottom line here is you need a lot of trials to be able to predict behavior properly and especially on an individual basis. So to summarize this part, one can predict behavioral signatures in at least some subjects from most of the brain. This came as a big surprise to us, but it seems to be a really robust finding. The fact that you can do that is typically masked by the fact that we look at group level rather than individual level analysis. And also robust prediction requires behavior, requires a lot of trials. So it is 1:46, so I can stop here, or I can show you a couple more slides with artificial neural network results. So just let me know what you prefer.
So there is no question. I have two questions, but I can hold it until end if you want to continue. So right now, except me, there is no other question. So I would, if you like, we can continue.
Okay, so let me show you the rest of it. It's not going to be very long, and I think it's interesting and hopefully provocative results. So I have so far showed you, hopefully convinced you that there's a lot of idiosyncrasy that is very important, and that individual subjects, are meaningfully different from the group in many circumstances. And when you look at individuals, you get meaningful differences compared to when you look at the group. But the question is, where did this idiosyncrasy come from? What are the sources of idiosyncrasy? It's a really hard question to answer. There could be so many different things. But one approach that one can take is to use artificial neural networks. So I'm going to just show you just a tiny bit of pilot results that we have that are only about behavior, but we think that that approach may generalize to brain activations. So in this project, we're looking at 3 different artificial neural networks. We're calling them RTNet, CNet, and MSDNet. And these networks are quite different from normal networks in that they are not only trying to classify images, but they also doing this in time, so they're giving meaningful response times. The details here really don't matter, so I'm not going to go through any of these details. But I do want to mention this is work of my former graduate students Farshad Rafi and my current postdoc, Matt Shecker. So the approach here is we have a simple digit identification task. People fixate in the middle, then they see a noisy image. Then they need to classify it as 1 through 8, and then give a confidence rating. We tested 60 human subjects. And we also had 3 different ANN architectures, artificial neural network architectures. And what we did was we trained 60 different instances for each architecture, with each of these instances only different in its random initializations. So nothing else is different. It's only its random initialization that is different. It is trained the same way. It is trained to the same criterion. It can do the primary task in the exact same way, all of these 60 instances, but yet we're going to look at where there's some individual difference between them. So as you can imagine, given that I'm talking about this, we find pretty large individual variability in both humans and ANNs. So this is the human data. We have 4 different conditions, easy and difficult, and also accuracy and speed focus. It doesn't really matter very much what the 4 conditions are. I just want you to focus on the fact that when you plot individual subjects, there's a lot of variability. And we all know that is just what human data looks like. But when you plot the accuracy for this RTNet network, CNET, MSDNet, this variability is quite substantial for each of these networks. This is for percent correct or accuracy. The same thing happens when we get these networks to generate reaction times. We again see a lot of variability between the different networks. Not just that, we can look at all kinds of different things. We can look at the skew of the reaction time distributions. Again, a lot of variability. We can look at reaction time for error and for correct trials separately, a lot of variability. We can look at confidence for error trials and correct trials, and again, a lot of variability, both for humans, but also for these networks. So we're quite excited about these results. We think that what this means is that ANNs may be able to serve as models for human idiosyncrasy. So this is some initial, very early evidence that random initialization alone can lead to large behavioral idiosyncrasy in artificial neural networks. We're testing that in a much wider range of neural networks. So these 3 were very specific. They're reaction time neural networks. This is not the standard type of neural network out there. But so far, we're seeing the exact same results in any neural network that we have tested. So this suggests perhaps that it is the different initializations with which we were born, the differences at birth in our brains that are leading to idiosyncrasy. That it's not necessarily our experience, or at least that even if our experiences were the same, we would still be seeing differences between subjects. And importantly, it is not necessarily just a cognitive bias. So all of these networks have the exact same decision-making strategies. So it is not a cognitive difference between them. It is just literally the weights that were used, the random weights that they were initialized with, that make all of the differences in the idiosyncrasy between them. So of course, this is a hypothesis for what may be happening in humans. We don't know. The fact that it happens in artificial neural networks does not mean that this necessarily happens in humans. We need to test it, and I don't know how to test that. But if you have ideas, please let me know. I think this is a really interesting hypothesis that I think we just need to figure out how to test. And I'm showing you using ANNs as models for human idiosyncrasy in behavior. What we also want to do, and we haven't done that yet, is using ANNs as a model for idiosyncrasy in brain activations. So again, that is still an open question of how exactly to do that. Okay, so just high-level conclusions. Again, the most important part here is that we have this simple computational model that is literally really simple. You can use it yourself. We have shared all of our data and code with on OSF, and it's easy to use. So you can take any maps that you have, just divide it into odd and even trials, and then using our model, you can estimate group versus subject level factors. We find lots of idiosyncrasy in brain maps, and the extent of that depends on the type of analysis. We can predict behavior for at least some subjects from almost anywhere in the brain, again, because of the idiosyncrasy. And we think that ANNs, artificial neural networks, are a promising tool for understanding human idiosyncrasy. Obviously, the jury is still out on this. So I want to thank my lab, especially the people who contributed to these studies that I showed you, our funding sources, and thank you for your attention.
Thank you so much. I appreciate it. It's a great talk. Thank you. So I would share a question. If you want to ask any question yourself, please let me know and I will unmute you. So the first question is from Cyrus. So you show effect of spatial smoothing on the reliability, but he's asking, are your results different if you use a smooth image? If you smooth the image basically more, if they smooth or less. So it's asking for the effect of a smoothness across different subjects.
Yeah, so smoothing has a little bit of an effect. Let me just go back to the results. So when you smooth more, you get more reliable and more consistent results. So here's the data. So when you look at, for example, the 5 mm smoothing, you tend to see slightly larger ratio for the subject to group, and the more smoothing you do, the less difference there is between the subject and the group level factors. So yes, there is a meaningful difference with smoothing. So the more smoothing you see, the more smoothing you do, the more similar people become. But even with a lot of smoothing, I mean, 20 millimeters is just a ton of smoothing. You still see that it is at least a 1-to-1 ratio for subject level factors to group level factors. So this does not disappear with a lot of smoothing.
Thank you. And Brad is asking, even if you use different initial parameter, model parameter for your artificial neural network, did you control for the variation optimization, like for example, for stochastic gradient descent or not?
So what we are doing in what we have been doing so far is we just train on the task until a criterion, which is which is sort of the normal thing people do. So we show them the same kind of images, and we train the network until it reaches 98% accuracy on the task. So that means that some networks are taking a little bit longer to get there than others. So they are seeing more examples. As a follow-up, we will be doing, we'll be fixing the epochs of training, so that every network sees the exact same number of images. That means, of course, that they won't reach the exact same criterion for accuracy. So you can have it both ways, unfortunately. We think we're going to see the same results, but we haven't done that yet. But yeah, so far, the training is identical in that it is the same process and it reaches the same threshold, but some networks see more images than others. We also need to actually correlate the number of images that they see with the idiosyncrasy later. My intuition is that it won't actually make that much of a difference, but I don't know for sure.
Thank you. And Luis is asking, what was the rationale or reason behind excluding the number digit 0 and 9 in your analysis?
Yeah, good question. So the reason was very practical. We wanted people to be able to respond quickly. So we're collecting reaction time data in this specific experiment, and reaction time was just very important. And it's pretty easy to use your 8 fingers and, to put it on 8 digits and respond pretty quickly. Once you add the thumb, it gets just not comfortable, and your reaction time is going to be slower for some keys. So we said 8 is enough. But yeah, it purely because we wanted good reaction time data.
Thank you. And a question for myself. So I appreciate the fact that you look at the effect of smoothing and also look at the 10% top n%. But I was wondering if you also look at the effect of the time. You have some result for one block and versus like more epoch, but have you look at the effect of the time, the data, that amount of data that you are using? Because we have seen the effect to some degree in the resting state.
What do you exactly mean of the amount of data?
So for example, if you have let's say, for example, 1 minute of the task, or versus 2 minutes of task, or like, I don't know, 2.5.
I see. Yeah.
And the reliability.
Yeah. All of the results I showed you is from all of the data. So in that sort of weird block level analysis, we
Mhm.
also looked at two blocks,
Mhm.
but that was sort of very, very specific.
Mhm.
The only thing where we looked a little bit more at that is this analysis, but that's a very specific analysis because we are again using 0700 trials. We're just changing how much you train on and how much you test on.
Mhm. Mhm.
But we're not throwing away any trials. We're always using 0700.
Yeah, I'm asking because we have like some interest in design rest, and also you show some evidence of the huge effect of noise. So I'm wondering like if there is like a um, sa saturated effect or not,
Yeah,
basically.
that's a good point.
And
Yeah,
yeah,
we
yeah,
can look at
and
that. We haven't done it. Yeah.
nice. And I have a question for me. So we are talking about the subject differences, right, between subject differences and subject variability, but also because this is my research area, it's like within subject variability. So I'm wondering if you think we should expect within subject similarity, that within similarity reliability is a good measurement, or the response, the brain response or activation pattern should be different from one trial and other trial or not.
Yeah, no, this is a good question. I think in our analysis, the extent to which one trial was different from a different trial, we just call it noise, right?
Mhm.
Which, of course, some of it is noise and a lot of it is not noise. It's just other variables that we're not measuring and we're not putting in the model. Um, so I assume that if you're able to control for more factors, then noise will become smaller and smaller. But yeah, I think this is an important question here is how much can you decrease that noise? Because right now, you have to go back to the to that result. Sorry, this is for the most standard analysis, and the total weight of the noise is still, even for 20 millimeter smoothing, it is more than 50%. If you add more things, more factors, like maybe you also look at accuracy, maybe you look at you measure pupil dilation, you measure skin conductance, you measure other things that also contribute to activations. Maybe you get the noise to decrease some more, but
Thank you. Yeah. Yeah, maybe we can follow up on that after the talk. So just talk about what we can do here, because that's an area that I'm also very interested. Uh, I know we are at 2 p.m., so I'm going to ask two quick questions, and hopefully we can follow up later on the Slack. Brad has a follow-up question that there are also many ways to randomly initialize the parameter and look at the effect of the individual. Is asking if is something that you are planning to explore or not.
Yeah, I would like to hear more. I mean, I think we just anytime you create a neural network, in the software that we're using um, sorry, I'm blanking on the name. It comes with random initialization. We didn't do anything special to the random initialization, but yeah, it's a good point that if we play with what that random initialization is, maybe we can learn more. So yeah, I'd be curious to learn more about that.
So I'm sure now Dr. Baker would love to chat with you about that. So a final question from Luis again is asking, are RT dependent on which digit is being activated, or is the interest in average RT or not?
Yeah, so we only look at average RT, and I'm sure that there's meaningful differences between the digits. This is not the kind of data that we usually collect in the lab because it is so uncontrolled. Some digits will be easier than others to perceive. I don't think that the accuracy is going to be exactly matched for all the digits. I don't think that RT is going to be matched. So yeah, so we're only focusing on average, but we did more analysis in the paper. This is a bioarchive version if you're interested, on the type of errors that we get for different images. I don't think we ever did an analysis that just looks at all of the ones versus all of the twos versus all of the threes, but we did an analysis for like each individual image separately from all of the others. But yeah, I'm sure there's substantial, or at least not insubstantial, differences in RT and accuracy and confidence between the different digits.
Thank you. Thank you so much for your time. I appreciate your talk. It was very interesting. I learned a lot. And thank you everyone for joining. I hope everyone have a wonderful Friday and weekend. Thank you.
Thank you.
Bye-bye.